Good evening, everyone. Welcome in yet another class of biochemistry, and we are going to discuss about protein folding in today's class. Okay, so this is our topic in today's class, and a very warm welcome to everyone. And let's crack NEET PG together. So, as I said, today we are going to discuss about protein folding. But before beginning with today's topic, let us have a quick introduction about the Unacademy. So, Unacademy is one of the largest online platform for the preparation of NEET PG examination, and you will get the best educators all over India at Unacademy. So, if anyone is planning for a online coaching class, then choose Unacademy as you will be mentored by the best educators. Unacademy is definitely special, and there are special classes at Unacademy. So what is so special about these special classes is that in special classes you can ask your doubts and queries directly with your faculty, and they'll be very much willing to answer your doubts. So it is like one-to-one kind of interaction. You won't feel anything different than a normal classroom like teaching. Then, as I always say, it is very important to attempt the MCQs and PYQs before going for any kind of competitive examination. So, Unacademy makes it possible by arranging test series and free tests. So, you can just go there and evaluate yourself how much you have learned so far. So, one of the benefit with Unacademy is live test series, like right, then it is very easy to start the process of learning. Just you can go to Play Store and type Unacademy, so you will see a logo of Unacademy learning app. When you will see the logo of an Academy learning app, you can just download the app and make your account, and you are good to go and start your preparation. And Unacademy, there are some of the very much features which are included with Unacademy. So, what are these features? Is flexible schedule. The schedule will be very much flexible. If the video is recorded, you can anytime go through that video if you are not particularly available at the time when it is going live. So flexibility of schedule, and very much there will be a simplified process of learning because you won't have to travel to some place. So time management will be quite very good. Then language of your choice, there are many options. You can learn in various kind of languages because many educators, they are making it possible to teach in various regional languages. So language can be of your choice. Then once if the video is recorded, you can go through that video as many number of times as you want to, and you can go through the quizzes which were once taken. You can retake the quizzes. So these all things make it very much interesting and very much interactive platform for the preparation. Then there are many students who feel that it is different from a normal classroom kind of teaching, but let me make it very clear that it is almost like classroom like teaching because you can chat with your educator. They'll be very much solving your doubts. Then there are interactive polls. Through these interactive polls, you will be able to attempt the questions, and you can understand that how many students they are getting confused in the same question. So interactive polls are there. Then lastly, but obvious, you can learn anytime, anywhere with the help of Unacademy learning platform. Then with Unacademy, there is another feature associated, that is Plus platform. So for this Plus platform or Plus subscription, you need to pay a little pocket-friendly amount that will be quite very less. So just paying this amount, you will be eligible for all the 19 subjects, and all the chapters and all the topics sequentially are available on the Plus platform. So Plus platform can be highly beneficial for a sequential studies. And there you can get updates about all the short-term courses which are conducted by the faculties, and crash courses for any kind of upcoming exam. So this all is possible with the help of Plus platform. So this plus platform, as I told, that the amount that you need to pay is quite very less. But it is my personal suggestion that go for a 12 monthly or a 24 monthly subscription, because this will be highly beneficial, as you can make the most out of all of the lectures which are pre-recorded. So there are immense videos. There are many faculties who are teaching the same topic. So whichever you find more comfortable, you can just check it out, and you can just grasp as much as you can. So just go for a yearly or a 2 yearly program and make the most out of it. And in this plus platform, still you can get a 10% additional discount if you're using my referral code, that is FB01. So by using my referral code FB01, you will be highly eligible for getting an additional 10% discount. So don't forget my referral code, that is FB01. So let's begin with today's topic, that is protein folding. It is one of the very interesting topic, and it is very important according to the aims and PGI exam point of view. Very many questions are asked on this. So let's begin with protein folding. What is our topic of today? Protein and folding. So how the proteins they are formed basically. Let's just revise the central dogma. Central dogma says that DNA will form RNA, and this RNA will form protein. This is what central dogma says. So the process of formation of RNA from DNA is called as transcription, and the formation of protein from RNA is called as translation. So just after translation, the protein that is formed is not your active protein. It is just a kind of polypeptide chain. And polypeptide chain is not directly eligible for performing all the functions which are allotted to that sequence. It's just a sequence which is randomly lying. It has got no use if it is not properly folded. So protein folding is everything when it comes to phenotypic expression of a particular kind of gene. If protein is being formed, it means, okay, fine, that gene is working properly. But if that protein does not attain a proper structure, a three-dimensional structure, it won't be of any use. So protein folding is what makes the main difference, or it is what most important in its action. So proteins are conformationally dynamic molecules. What do we mean by dynamic? That the folding of protein, if it is folded in a specific type, then it can change the type of folding pattern. It is dynamic, conformationally dynamic molecule that can fold into functionally competent conformation, and that can change into competent conformation. It can change into a type of structure which is of some kind of use, functionally competent. So just highlight this, functionally competent conformation. This is what we are going to see in protein folding. Protein folding actually leads to formation of functionally competent conformation. Now, occupying a specific 3D conformation, occupying a specific 3D conformation is what called as protein folding. Process of incorporating specific conformation for protein is called as protein folding. So it is the 3D structure which actually refers to the activity of protein, right? Let's move further. These proteins, they are folded with the help of accessory protein. So in the last slide, we just missed the line. So when we are saying protein folding, so protein folding, how will it get folded? If it is made directly, how will it be folded? So there are some accessory or auxiliary proteins which are actually playing very important role in folding of protein, these auxiliary proteins. This auxiliary proteins, what is their specific name? What is their specific kind? These auxiliary proteins, they are specifically called as chaperones. Remember this word, chaperones. So they are the assistant in protein folding, right? So protein folding, we know why it is important. What happens in protein folding, that we are going to see in detail, but we know If there is a polypeptide chain, just a sequence of amino acid arranged sequentially, it needs to fold on itself for becoming active. So that is your protein folding, right? And the things which help in protein folding, the most important one is chaperones. So we are going to start our lecture with the reference of chaperones. So what are these chaperones? What are the properties of these chaperones? What are they actually in nature when we are saying chaperones? So let's begin with the idea of chaperones. Let me take pen. Yeah. So what are these chaperones? They are present in wide range of species from bacteria to humans. So they are present in bacteria as well as they are present in humans, right? These chaperones. Now, chaperones, they are also called as heat shock proteins. Their name is heat shock proteins. Another name is heat shock proteins. What do we mean by heat shock proteins? If we are giving it a word, that heat shock proteins, it means that they are inducible or they can get converted into their actual function. They can start playing their actual function, or they are inducible by conditions that cause unfolding of newly synthesized proteins. Example, when there is elevated temperature or there are certain kind of chemicals, these heat shock proteins, they will start acting and they will just rescue the molecules of proteins which are on the verge of unfolding or denaturation. So heat shock proteins, like, just let us take a pen and let us start understanding this in detail. If this is a kind of protein and there is a high temperature, so this protein will start changing into some other form, like this. So the nature doesn't want it to be changed into useful information, in an information which is not of further use. So what will happen? There will be another molecule, that is your chaperones. It will be lying somewhere in the environment, and as soon as it is starting to denature or change its state, it will just engulf it into itself. Will engulf it into itself, and when it will engulf it, it will save it from denaturation. So this is heat shock proteins, where whenever there will be elevation in the temperature or there will be elevation in the chemicals, will lead into activation of heat shock proteins, which are called as chaperones. They bind to predominantly hydrophobic regions of unfolded proteins and prevent their aggregation. Where does they bind? They bind at the hydrophobic regions. So whenever we are talking about any particular kind of protein, the protein will be formed up of hydrophobic regions and hydrophilic regions. Hydrophilic regions will be lying on the surface of that protein. The surface of the protein will be made up of hydrophilic molecules, and the interior will be made up of hydrophobic, and the exterior will be formed up of hydrophilic molecules. So the chaperones, what they will do, they will bind to the hydrophobic ends and it will save the protein from aggregation or changing into its denatured form. So editing mechanism in detecting misfolded or defective proteins. As soon as there is change in the environment, the chaperones, they will just come to the rescue of the proteins and it will try to save the proteins from that unwanted situation, right? It helps protein takes its functional form. And if the protein is just now formed, if we are not talking about its heat shock nature, then if the protein it is just now formed, what will it do? It will take up this protein inside itself. We'll be just seeing the exact structure of chaperone also. So it will take the protein inside and it will change this protein into its activated form. So this is the function of chaperone. So remember the word very well, chaperones. So they act in part as the quality control or editing mechanism for detecting the misfolded or otherwise defective proteins. What is its function? We have just seen in the last slide that detecting the misfolded or otherwise defective proteins. There will be a change in the environment and it will get activated and it will detect these misfolded or defective proteins and it will take them inside and it will start functioning. Now, if we are talking about chaperones, that they are so important, so what we need to know, whether the activity of chaperone is ATP dependent or it is not ATP dependent. So the activity of chaperones in most of the cases is ATP dependent. So wherever I am highlighting the things, you just try to keep at least these things in your mind, because they are very much important for the PYQ. They are very much important for the questions. Like wherever I am highlighting, they are some or the other time asked in the exams. So this chaperone, most of the time its activity is ATP dependent. It's ATPase activity, right? It will be assisted by ATPs, and it itself behaves like an ATPase, right? Let's move further. So these chaperones, they belong to various categories, or there is a group of chaperones. They are not of a single type. They are of various types. So let us just discuss some of the examples of molecular chaperones. They are HSP 70, heat shock protein 70, heat shock protein 90, heat shock protein 40, BIP, immunoglobulin heavy chain binding protein, BIP, glucogen regulated protein, GRP 94, calreticulin and calnaxin. These all are some of the examples of molecular chaperones. So in this slide, I have included the names of all those chaperones which are important. So all the mechanism you not need to know, because that is not very much important according to the exam. In exam, they will be mostly asking you that amongst the following, which one is the example of a molecular chaperone. So what you can do is you can just remember the names I have mentioned over here. That is BIP, GRP, and calreticulin and calnaxin. So you'll be able to identify which one among the following chemical compounds or molecular compound is belonging to the category of molecular chaperone, right? Okay, let's move further. So there are enzymes which are actually participating in the proper folding of these proteins. So these enzymes which are actually participating in the proper folding of proteins, they are of two types, that is PDI, that is your PDI, or protein disulfide isomerase, or PPI, that is peptidyl prolyl isomerase. These two are the enzymes which are actually participating in the process of proper protein folding, right? Now, this we have seen that enzyme assisted folding, there are the two types of enzyme that are participating, that is protein disulfide isomerase and peptidyl prolyl isomerase. So try to remember the names, right? Now there are another type of chaperones which are given very much importance, that is chaperonins. So what are these chaperonins? Chaperonins, they are actually another class of chaperones. They are chaperones only, but they are of another class, chaperones. And the chaperones, when we are saying that they belong to the category of chaperonins only, so they are HSP 60 family of chaperones. HSP 60 makes the chaperonins family of, sometimes they are called, and if it is called as HSP 60, it means it is called as chaperones, and when HSP 60, its synonym is chaperonins as well, right? So what are these chaperonins? They are barrel-like structure. Their structure is barrel-like or a drum-like kind of drum in which unfolded protein is retained. The unfolded protein will just go inside this drum, and after some time it will be given all the possible suitable conditions, and now it will be changing into a properly folded protein, folded properly, right? So one of the example of the chaperonins is empty grow L or empty grow EL, chaperonin, right? So in just next slide, we'll be seeing how this chaperonin will actually work. So here, this is your grow EL, and in this, the protein, which is just a polypeptide for now, enters inside the barrel. So this is two-layered barrel, and when it enters inside the barrel, it's an ATP assisted process. Now, when ATP enters, there will be a cap which will be added on the top of GRO-EL. GRO-EL is the barrel and the lid of GRO-EL is called as GRO-ES. The lid of the GRO-EL will come and bind over here. So now inside this, what all things are inside this is your protein and your ATP. This barrel contains protein as well as ATP, and it is an ATP dependent process. So what will happen? There will be release of ADP, an inorganic phosphate, and the lid, that is GRO-ES, will also be removed after some time, and properly folded protein will come out of the barrel, that is your GRO-EL or your chaperonin. So this is how the process of proper folding occurs in chaperonin or chaperonin dependent proper protein folding. This we have seen here, right? So let's move further. We have understood about the enzymes which are responsible for proper folding. Then we have understood about chaperones and then chaperonins. And now let us move further, that how the endoplasmic reticulum is important in proper protein folding. Is there any function of endoplasmic reticulum in the proper folding of protein? So we'll be just seeing that in this slide, right? So role of endoplasmic reticulum in proper protein folding. The endoplasmic reticulum function as the quality control compartment of the cell. So it will just see whether the proper folding of protein has occurred or not. So you can just highlight as the quality control department, or it will just check whether the proteins are properly folded or not. The newly synthesized protein after entering the ER fold. So when they are newly synthesized, they after entering into the ER fold with the help of chaperones, the folding enzymes in the lumen of ER. With the help of chaperones, it will enter inside and it will further just change into the properly folded protein. Now, endoplasmic reticulum stress sensors initiate the unfolded protein response. So what happens is that mechanism to sense the levels of misfolded proteins and initiate the intracellular signal mechanism to compensate for the stress condition and restore ER homeostasis is unfolded protein response. If the proteins are not properly folded, so is there any mechanism to just check out whether the proteins they are folded properly or not, or is there any rescue mechanism so that now your cell does not contain the improperly folded protein? So that all occurs in your ER. So the mechanism to sense that, the mechanism to understand, or to mechanism to identify the misfolded proteins is done by ER. So what happens is intracellular signaling will start and it will be identified as a stress condition, and when there will be a proper stress condition identified, ER will just restore the whole process and it will try to manage the conditions in the cell, that is ER will just maintain the homeostasis. So unfolded protein response, the process which is initiated after the improper folded protein has been formed, it is called as unfolded protein response. Unfolded protein response. So unfolded protein increases the ER folding capacity. So whenever the cell will sense the unfolded protein response, what it will do? It will just empower endoplasmic reticulum with higher fold of folding capacity, and it will just save the cell from the building up of improperly folded protein, because improperly folded proteins, they are highly toxic for the cells. So now they won't be made when, or they won't be let in the environment for a longer time. So ER is kind of a police for identifying the improperly folded protein, or the proteins which are not conducting their duty in a right manner. So they will be just checked by the ER, right? Now, there is another thing, that is ERAD. So if they are not folded properly and they are not even improving, so what cell will do? Cell will definitely try to stop its activity. And if it has stopped its activity, it means that they are no longer needed in the environment. So if they are no longer needed in the environment, it means that they need to be degraded. So who is actually just identifying this improperly folded proteins? We are saying that ER, endoplasmic reticulum, is identifying the unfolded protein. So ER will only further lead degradation of the misfolded protein. Like police, they are identifying the people who are not performing their duties in a proper way, or a traffic police, he is not, he has identified the people who are just violating the traffic rules. So now further they will just put the fine on them, or they will do the further process of punishing them, like whatever kind of charges they will try to put if they are not wearing a helmet, or they are just tripling on the bike. So they will further take the charge. So endoplasmic reticulum will further take the charge, and it will take the charge in what manner? It will start degrading these misfolded proteins. So it is also called as ERAD, endoplasmic reticulum associated degradation. So misfolded or incompletely folded proteins interact with chaperones, which retain them in the ER and prevent them from being exported to their final destination. So if uh, they are not properly functioning, so they won't be transported, or they won't be taken to the place where their function is. They won't be exported to their final destination. But obvious, because they are not doing their work properly, or they are not properly folded, so how would they be capable of performing the further duties? So they need to be stopped there only. So the misfolded proteins are usually disposed by ERAD. So usually they are disposed by ERAD, endoplasmic reticulum associated degradation, or endoplasmic reticulum associated protein degradation. Many a times in the text it is also written like protein degradation, ERAD, but in the short form they are not highlighting the protein, right? So let's move further. Protein degradation. So we have moved from protein folding to protein degradation. We have seen that how the protein, they are properly folded with the help of chaperones and chaperones. Then we have seen that if they are not properly folded, so who will take the charge in the cell that they are not properly folded? So endoplasmic reticulum takes the charge that now they are not properly folded. So they will identify the misfolded proteins. And when they have identified the misfolded proteins, their final fate is degradation. So let us move to protein degradation only in case of misfolded proteins, right? So let's move to misfolded protein. So now when your misfolded proteins are formed, they cannot be let into the environment for a long duration. They cannot be left out for, okay, fine, you remain there and someone will come and will try to erase you or try to degrade you. The things, they need to be done very fast, because if the misfolded proteins, they remain in the environment, they'll be highly toxic for the environment, because there are many disorders and many diseases which are because of the misfolded protein. So they need to be cleared up very fast. So one of the things is that misfolded proteins should be rapidly degraded. Rapidly degraded. Rapidly degraded. Right? So rapidly degraded, it means, what does this rapid degradation means? Whenever a protein is formed, it has its own half-life. So its half-life will decrease. Its half-life will decrease when it is improperly folded. So intracellular proteases hydrolyze the internal peptide bonds. So how the process of degradation of protein will occur is intracellular proteases, they will hydrolyze the internal peptide bonds. It won't start cleaving the outside peptide bonds, but it will start cleaving the internal peptide bonds, because internal peptide bonds, if they are cleaved, it means it will be rapidly degraded. So that's why specifically here internal peptide bonds is highlighted, because they will lead into non-functional protein very fast, right? The resulting peptides are then degraded into amino acids, and when they are degraded, when there is a cut in the internal bonds of the long polypeptide, like if it's a 200 amino acid long polypeptide, there will be a cut at somewhere 90, 100, 120 like this internal peptide. So when there will be a cut, it will lead into formation of degraded amino acid or amino acids which are free in the environment by endopeptidases that cleave the internal bond. So the enzyme is endopeptidase. So simply the meaning of endopeptidase is it is cleaving the internal bonds, right? It is cleaving the internal bonds. So endopeptidase by aminopeptidases and carboxypeptidases that remove amino acid sequentially from the amino terminal and carboxy terminal respectively. So endopeptidases, they will cleave the internal bonds, and now when they need to uh, cleave the smaller polypeptides also into smaller amino acid or dipeptides, tripeptides, or tetra or heptapeptides, which enzymes will take up the process? Aminopeptidases and carboxypeptidases. Aminopeptidases, it will start cleaving the chain from the amino terminal, and carboxypeptidases, it will start cleaving the chain from the carboxy terminal, right? So this is your protein degradation, internal degradation, and when internal degradation has occurred, the amino acid should be freed out. So that occurs with the help of aminopeptidases and carboxypeptidases, right? Then, is there any specific sequence where the degradation will occur? So the sequence name is PEST for remembering. PEST sequences. Let's take another. Are the regions which are rich in proline, P for proline, the sequences which are rich are proline, glutamate, that is denoted by E, serine, S, and threonine by T. Target some of the proteins for rapid degradation. If there are these sequences, proline, glutamate, serine, and threonine, it will just lead into rapid degradation. It will lead into a very quick degradation. And here you can see P is for proline, E is for glutamate, S is for serine, and T is for threonine. So in one of my another lecture where I have done classification and structure of amino acid, I have explained clearly how to name the amino acids. So there is a very nice mnemonic. You can just go and check out that video and you will be able to remember the initials of each and every amino acid for your exam point of view and for further reference also. So PEST, proline, glutamate, serine, and threonine. Right? Let's move further. So now the protein degradation, which we are saying that there will be degradation of proteins will be degraded. So proteins can be degraded by two ways, that is at ATP independent degradation, and there will be ATP independent and ATP dependent degradation. So first of all, we are going to see ATP independent degradation. What happens in ATP independent degradation? So if we are saying it is ATP independent degradation, it means that there will be no use of ATP. ATP independent. Protein that undergo ATP independent degradation without the use of ATP. Which type of proteins they are? They are extracellular proteins. They are membrane associated proteins. And they are long-lived intracellular proteins. The proteins which are once formed and they are required in the cells in the intracellular environment for a very long duration. They are called as long-lived intracellular proteins. So proteins which will undergo independent degradation, the site will be lysosome. The site where this process of ATP independent degradation occurs is your lysosome by ATP independent mechanism, and one of the example is blood glycoprotein. It undergoes ATP independent degradation in lysosome and it is a kind of extracellular protein. It is membrane associated and it is a long-lived intracellular protein. It is required for a very long duration inside the cell, right? So ATP independent degradation we have seen. Then another thing that we need to see is ATP dependent degradation. ATP dependent. So it will be like with the help of ATP, at the expenditure of ATP, there will be a usage of ATP. So what kind of proteins that undergo ATP dependent degradation? They are the regulatory proteins. The regulatory proteins with short half-lives. The proteins which have got very short half-life will undergo ATP dependent degradation, and abnormal or misfolded proteins, they will undergo ATP dependent degradation, right? Abnormal or misfolded proteins. So the process of ATP dependent degradation, it occurs in a special place, that is your proteasomal complex, that is in the cytosol only, and it is proteasomal complex, right? Now ATP dependent mechanism, it is further extended with the help of another kind of molecule that is called as ubiquitin, right? So we are going to see this mechanism of ubiquitin dependent protein degradation now further, and it is a part of ATP dependent degradation, and this ATP dependent degradation is also called as endoplasmic reticulum associated degradation, and it is with the help of ubiquitin. So we will be seeing the mechanism that how this ubiquitin will be further actually contributing in the cleavage of misfolded proteins. So first of all, let us have a look here. So endoplasmic reticulum associated degradation. So we said that it is with the help of proteasome. So peptides, the peptide or the protein, if it is misfolded, it will bind with ubiquitin. So here can you see the ubiquitin quite clearly? This is a single molecule of ubiquitin, and there are 1, 2, 3 and 4 molecules of ubiquitin getting attached with the protein which needs to be degraded, right? And finally, it will be entering into a proteasome assembly for its further degradation. So this is endoplasmic reticulum, and we clearly talked about that which molecule first of all identify that yes, there is a misfolded protein. So your endoplasmic reticulum is the thing which identifies the misfolded protein, and it identified it, it engulfed it, and from its lumen, it just released the target protein. Now it is called as target protein. Target word over here is for degradation. Degradation or the misfolded protein. Misfolded protein or target protein or protein which is now destined to degradation. It is released in the environment, and when it is released in the environment, or we can say cytosol. Just a second, let me take the slide where we were just talking about it. So we were, oh God, just a second, there is some issue. Just a second, there is some issue. Hmm. So we were over here, right? So the proteins which are now in the environment, intracellular, in the intracellular environment, that is your target protein. And target, why? Because it is misfolded and it is now destined to degradation. It will join with certain ubiquitin, like over here you can see 4 ubiquitin molecules are binded at the end. When they are binded at the end, it will be further giving the signal that yes, now they need to be cleaved, and they will enter into a proteasome assembly and they will be further degraded, right? Now the steps of endoplasmic reticulum associated protein degradation. So how it occurs? A target protein which is misfolded undergoes a retrograde transport through the ER membrane into the cytosol. It is actually destined to release in the outer environment, but now it will be again released into the internal environment only. So it is called as retrotranslocation or dislocation into the cytosol. And when it is released in the cytosol, we just saw in the picture that how it is polyubiquitination occurs, that there will be addition of many ubiquitin residues at the end of the polypeptide. So When there will be so many ubiquitin, like minimum there should be 4, there can be many ubiquitins. So when it is attached to your polypeptide, will enter into a proteasome, inside which it is degraded into small polypeptides that exit and may have several fates. So when it will be degraded, it will be having several fates. So many things can occur with it. So what is this ubiquitin? We must know about ubiquitin. So it is a key molecule in protein degradation. You should remember that it is the key molecule. This is very important for PYQ. And how many amino acid it is made up of? It's a very small protein. It is made up of just 76 amino acid. It's a very small one. So 76 amino acid long it is, right? Then it is highly conserved protein. It is almost same in literally a range of organisms. It is highly conserved protein. So attachment of protein and ubiquitin is called as kiss of death. So if protein it is attached with the ubiquitin and at least 4 ubiquitin molecule, it means now it is destined to death or destined to degradation. So that's why it is called as kiss of death. Okay, minimum of 4 ubiquitin molecules are needed to degrade one molecule of protein. So we have seen it through picture also that at least 4 ubiquitin molecules are needed. And also remember what is this kiss of death, because many a times there will be a MCQ. What is kiss of death? If your protein is into ERAD or your protein is into ATP independent mechanism degradation, or it is to ubiquitin or none of the above. So your answer would be when it joins with ubiquitin, it means now it has kissed the death and it is now destined for further degradation. There will be no excuse in its degradation. It will degrade, right? Now there is an N end rule for ubiquitin binding. And what is this N end rule for ubiquitin binding? There is a rule. Ubiquitin binds protein with PEST. So we have seen PEST previously also, that it stands for proline, glutamate, serine and threonine. So the sequence is very easy for you to learn. Like P, you will be very easily remembering proline, and S for serine and T for threonine, just E for glutamate or glutamic acid, you can remember very well. So ubiquitin, it will be binding at these amino acids. So there can be questions of various series, proline, glutamine, serine and glutamic acid and serine and threonine, or there will be some other combination. So you have to remember this code PEST, or it can be shuffled also. So if you are remembering this thing, remember it well, that ubiquitin, it will be binding at the N terminal where it can find proline, glutamic acid, serine and threonine, and it will be binding to it. Right? Now we have understood what is ubiquitin, we have understood what is endoplasmic reticulum, and lastly in the degradation series there is a proteasome. So what is this proteasome? Ubiquitinated proteins enter into proteasome assembly, and where is this located? It is located in the cytosol, and it is a large cylindrical structure of 50 subunit. It is a large structure, and it is also a barrel shaped, or it is cylindrical in nature, or it is a trunk or a drum shaped, and it is also a very small molecule, not a very big molecule. It is, oh sorry, it's a very big molecule, sorry, it's a very big molecule made up of 50 subunits. Small was your ubiquitin. Here it's a very big molecule made up of 50 subunits, and it is an ATP dependent process. So we are reading this whole thing ERAD in our example of ATP dependent degradation. So it is ATP dependent process, right? Let's move further. We have seen proteasome. After ubiquitination, it needs to degrade. So this degradation occurs in the proteasome assembly, and it is located in the cytoplasm. Now proteasome, they are protein complexes. They are made up of protein. It's a large cylindrical structure or drum or barrel shape. And it is made up of what? So it is made up of four rings with hollow core. It is made up of four rings with hollow core. See here, you can see the hollow containing the protease active site. So interior of this barrel contains the protease active site. One or two caps or regulatory particles that recognize the polyubiquitinated substance. Or and then it will have one or two caps which will be identifying the ubiquitin bases. So ubiquitin residues, because they need to be identified, because it is only occurring with the help of ubiquitin. Whichever thing will be ubiquitinated, it means now it is destined to further degradation. So it has got a regulatory particle which identifies or which recognizes the polyubiquitinated substrates, right? So this is the structure. See this, 1, 2, 3 and 4. 4 rings you can see. Then over here, this is your ubiquitin and this is your regulatory site, and regulatory site is binding the ubiquitin, and when it has identified the ubiquitin, it will engulf the whole protein inside. You can see that in the hollow barrel there is this structure of the protein which needs to be degraded is lying, and in the interior of the ring there is protease active site. So these protease active sites will further act upon this protein molecule and it will degrade it. So from one end it will enter and from the another end it will be released out, and both of them are called as regulatory particles, because when it is making the protein enter inside it, it is identifying the ubiquitin residues, and when it has broken down the protein, it needs to make sure that these proteins are now properly broken down or not, because if they are not properly broken down in the legitimate size, they can be of further harm. So this is how the proteasome assembly participate in degradation of the misfolded proteins. So it is made up of 50 subunits, right? So it's a very large molecule. Fine. Let's move further. So what are these steps? We have just discussed these steps. So just have a quick revision. The regulatory particle recognizes the ubiquitinated protein which are folded by ATPase present in the regulatory particles or caps. We just saw that how the regulatory particles, they have got ATPase activity and they will recognize and they will engulf the protein which is misfolded, and when it has engulfed it, it will have the protease active site inside itself which will perform the further degradation. So protease active site are present in the interior of the protease, and peptides are released into the cytosol for further degradation by cytosolic peptidase, but it needs to be degraded into further smaller polypeptides. So there are these regulatory sites present at both of the ends of your proteasome, so that they first, when it is entering, it is just identifying the ubiquitin residues, and when the degraded protein is coming out, it should be of a particular size only. So there are two regulatory units. Now coming to the last part of today's class, that is protein misfolding disorders. So from the starting we were saying that yes, protein misfolding is very dangerous for the cell. So how is it dangerous? What are the things that occur? So there will be prion diseases and there will be prion related protein diseases. These two things occur because of the protein misfolding, and we are going to see this in further slides, that how the protein misfolding is actually causing some kind of problems in the cell. It is called as prion disease and it is called as prion related protein diseases. So let us just have a look at this prion diseases and prion related protein diseases, right? So let us just start with this topic. So what are first of all prions? Due to let me take pen. Yes. Okay. So protein misfolding disorders. So because of the failure of properly folded protein, formation and it leads to rapid degradation. So half-life, we just discussed that half-life decreases. So half-life decreases, it means it is rapidly degrading. Rapidly degrading. Right? This is the thing. Now, what are prions? Because both of the things are prion diseases or prion related protein diseases. So what are these prions? Prions is accumulation of misfolded or partially degraded proteins. Right? What are prions? They accumulation of misfolded or partially misfolded or partially degraded proteins. So this is your prion. So let us see what are the diseases which are associated with prions. So let us see. Creutzfeldt-Jakob disease is because of the prion accumulation. Then GSS disease is because of prion accumulation. Then insomnia is because of prion accumulation. And the Kuru disease is because of prions. Right? So these four things you need to remember well. That is your Creutzfeldt-Jakob disease, your GSS disease, and insomnia and Kuru disease is because of the prions. So remember Creutzfeldt-Jakob, GSS, insomnia, and Kuru. Let's move towards the next thing, that is prion related protein disease. So what are prion related protein diseases? There are some examples. Alzheimer's disease, then Parkinson's disease, then beta thalassemia, ignore the spelling, beta thalassemia, and amyloidosis. So these are the prion related protein diseases. So diseases are very important because direct questions will be asked whether they belong to the prion diseases or prion related protein diseases. So you need to just identify the difference. So we'll just revise them once again. That is your prion diseases or prion related. So they are Creutzfeldt-Jakob disease, Creutzfeldt-Jakob disease, then it is your GSS disease, then it is insomnia, then it is Kuru disease because of the prions. Then prion related, what are the diseases which are related to prion related protein diseases? Alzheimer's, Parkinson's, beta thalassemia, and amyloidosis. So these are the things which you need to remember. So in today's class, we have tried to cover almost everything which is related to protein folding and protein misfolding and protein degradation. So we have seen so many things in the class. That is your proper protein folding, then your protein misfolding, then protein degradation, and lastly diseases related to improperly folded proteins. So we have seen proper protein folding, protein misfolding, protein degradation, and disease related to improper protein folding. We have seen what are chaperones, we have seen some of the examples, we have seen what is chaperonins, then we have seen the ERAD mechanism, endoplasmic related and endoplasmic reticulum associated protein degradation. Then we have seen what is ATP dependent degradation, that is your ERAD, and then finally what is ubiquitin and what is ubiquitination. Then we have seen what is this proteasome assembly, and last point with the proteasome, it just clicked, that is proteasome inhibitor. It's very important. So let me just write it over here with another color. That is your proteasome, proteasome inhibitor. Very important question for various exams, and it was asked in various exams. So what is proteasome inhibitor? It is bortezomib. Bortezomib is your protease inhibitor, and it is protease inhibitor. So what is its utility? It is used in multiple myeloma for hepatocellular carcinoma. So in various types of myelinomas, and with which type of carcinoma? Hepatocellular myelomas and hepatocellular carcinomas. So protease inhibitor, it is bortezomib, which is a very important molecule for treatment of various carcinomas. So we have tried to cover almost everything in today's class regarding protein folding, and that is it for today's class. And if you find my class useful, please give a like to my video, and also subscribe our channel, that is Let's Crack NEET PG. Hit the bell icon for getting all the notifications for the upcoming videos, and use my referral code, that is FB01, for getting an additional 10% discount in your Plus subscription. And if you are interested or you have some doubt in particular biochemistry topic, you can just write down in the comment section, and I'll be going through the comment and I'll be trying to make a video on that particular topic. So just go through the lecture well, learn the concepts, and try to solve the MCQs and PYQs related to the topic, and also comment what you want to read in further classes. This is it for today's class. Hope you have enjoyed the concept which I was trying to give you in today's class, and have a very good day. Happy studying.